Great. Thanks to the organizers for giving me this opportunity to discuss with you some of our kind of ideas in this space. And today I just want to give you a kind of a vignette of the types of things we're thinking about in terms of how do you engineer microbiomes towards potentially new metabolic functions or new capabilities and opportunities in which we can potentially take that into engineering mammalian genomes. So, in general, metabolism is really key to all processes in life. And as we know, kind of as we're learning more and more over the recent discoveries that aberrant pathways and metabolism can lead to very drastic diseases, things like cancer, where it's not just mutations that lead to propagations of cells, but mutations that lead to the support of aberrant metabolic processes that allow cancer cells to be able to grow and invade tissues. And that's also true in the context of understanding how we process natural foods and disorders associated with kind of food processing as is associated with things like the microbiome. So if you look at kind of the microbiome, and the gut microbiome in general, it's actually very interesting. It provides a lot of very essential metabolic capabilities in its ability to produce things like short-chain fatty acids, bile acids, a variety of small molecules and vitamins that are really crucial for the development and maintenance of health and disease, as well as lipids and processing of different amino acids. And I think this is also an opportunity where we can start to think about some of the synthesis approaches that we've been talking about. Jeff just mentioned a really great example of salmonella, which some of those sister genomes are present in the human genome or the human microbiome, at least. And so, this is where I think there's also kind of really important opportunities where we can not only think about engineering at the scale of individual bacterial genomes, but potentially populations of microbial genomes. To take amino acid as a really interesting example, so if you look at kind of mammalian cells, they really have the ability to process and generate only 11 of the 20 required amino acids, and the rest of them are really derived from diet. And so if you actually look at the phylogenetic tree, basically only plants and fungi and some of the bacteria species are able to produce all of the essential amino acids on Earth. And the rest of essentially the tree of life derives those amino acids by consuming plants and fungi and other organisms through the food web. And so, so in order for us to be able to survive kind of higher order organisms, you really need to get them from the diet. And there's a lot of really interesting historical reasons for why these things might be lost. These tend to be longer pathways that the longer the pathways are, the more likely it is they're somehow lost during the evolutionary process. Now, this has immediate applications in terms of thinking about utilizing these cells in a synthetic context. So, for example, if you want to grow human or various mammalian cells in a dish, you need to supply these essential metabolites, things like vitamins and amino acids. But in addition, you also need to supply other more complex substrates, things that are derived from things like serum, right? So essentially, all of the media are derived from some form of serum that comes from an animal. And this is potentially challenging and an important opportunity where we can start to think about some of these aspects where synthesis could help. But if you look at kind of bacteria, it's very interesting. They can basically survive off of very simple carbon sources and basically some elemental building blocks, and they produce a variety of different genotypes and phenotypes. And I think bacteria is a really interesting opportunity to start to model some of these things because you can actually create the auxotrophic phenotypes that we see in higher eukaryotes by essentially removing some of the genes associated, for example, with amino acid biosynthesis, such that they're no longer able to grow. but when you put them together in some synthetic context, they're able to survive. And this actually quite nicely recapitulates some of the things that you probably are seeing in your gut microbiota. So if you look at kind of metabolic exchange, there's kind of good ways in which we've systematically been exploring the how these bacteria crossfeed with one another. You can use that as a basis to start to model metabolic exchange in the microbial context, both analytically as well as experimentally. We've had a really fun collaboration with economists to talk about this in the context of what, of maybe microbial markets, right? So basically how microbes exchange metabolites are very similar, at least in principle and conceptually, as that of economic markets. And so we made good analogies and frameworks to start to study those things. And not only do microbes exchange metabolites, but they also exchange DNA. As Jeff had just alluded to, they're trying to, block the exchange of DNA. But I think this is also an opportunity where we can potentially exploit this for some of the metabolic engineering processes. For example, in this example, it's quite interesting that essentially there's been a process by which ocean bacteria transfer genes associated with the processing of seaweed into the gut microbiota of individuals, and in this case, in Japanese populations where they consume an abundance amount of these types of foods to benefit the host, right? And I think this is a kind of an elegant example of nature's ability to reprogram the microbiota. And so in our lab, we've been thinking about these opportunities, what I call mobiotics, where you can start to do gene therapy on whole microbial communities by introducing strains that could introduce specific pathways and genes that can propagate within this community in a natural kind of environment. I won't have too much time to show you, but this is just to show that we've done some of these experiments in animals and in mice, and we can see that we can transfer genetic material directly into those native microbiota in the mice, and they propagate over long periods of time. And some of these pathways are quite interesting because you can use them, for example, to start to complement some of these pathways that potentially are missing in mammalian systems. So this is, for example, a catalog of a set of genes that I had described in one of the previous genome project meetings about fixing some of the missing pathways in mammalian cells. And this really, I think, is an opportunity to start to synthesize not only kind of entire human genomes or mammalian genomes, but also hundreds of thousands of bacterial genomes, basically for the same kind of price and potentially effort. With that, I just want to kind of wrap up here and say that we're thinking about this in the context of potentially producing not just metabolic molecules, but also things like neurotransmitters that might affect additional host factors, things like cognition, memory, and things of that sort in the gut. And with that, we're now taking the steps into thinking about how we can introduce these pathways into mammalian cell lines, and this is a collaboration with Jeff Buka and NYU here, where we're trying to introduce the biosynthetic pathways for these missing amino acids back into mammalian cell lines. And we're happy to note that this effort is being supported by DARPA for this, and we're very happy to kind of share with you kind of future advances and kind of update you on the type of things that we're trying to do in this space. But really kind of the vision for this is that we think this is one way to try to disrupt some of the barriers associated with mammalian culture and production of biologics, and also thinking about this in the context of understanding mammalian metabolomes by generating cell lines that are able to self-sustain themselves in the laboratory. With that, I'd like to just acknowledge everyone in the lab, our collaborators, and a variety of funding sources. stunned into silence.
So,
Yes.
So which amino acids can the mammalian cells now? Which amino acids can the mammalian cells now make?
We're just starting those experiments, and so we'll let you know.
Okay, okay, and I guess this means you have to hand make the media.
Right. Yeah. So I think that's the really kind of where things. That's the starting point. We know that in the dropout medias they don't grow. So that's the clear control. And so the question is, if you add in these pathways, do they do they start growing? And so. Okay, I think we're gonna wrap up here. Thanks.